first time in my life, and I've been overwhelmed by the extraordinary hospitality that I've received. It was an immense pleasure to be able to visit this Yalu Gyesha primeval forest in the last 2 days, thanks most particularly to Toma Kamila Brzezinska, who showed me around, fascinating part of the country, and I'm really so very grateful to be given this opportunity to see this part of Poland. So this is the topic of my talk, and it's a special pleasure to show this first slide, which was taken a while back and recognizes the enormous contribution that Krzysztof Tarkowski has made. That's one of the most famous of all Polish embryologists. He and Aaron, shown here, really pioneered the whole principle of doing experimental embryology in mammals, because at that time nobody knew how to take the embryos out, do things with them experimentally, and then reintroduce them back to an animal. And of course, Krzysztof went on to do amazing experiments involving the recombination of embryonic cells and making mosaics and so many other things. So this is why it's a special pleasure for me to be introduced by him. Now, one of the principles of development is that we all start life as an egg, and gradually cells are formed, dividing as they go, but the general principle is that cells do not go back from one pathway to another. So you would not find that nerve can be transformed into heart or muscle into intestine. So first one might ask, how does this whole process of one cell, the egg cell, give rise to so many different cell types? And it turns out there are two fundamental principles involved in this. One is that the egg, the single starting cell, is heterogeneous. Not all parts of it are the same, and you see the part of the egg which is yellow goes into the yellow cells, and the part of the egg which is blue goes into the blue cells. So you have two different kinds of cells to start the whole process off. This principle has actually been known for quite a large number of years in the invertebrates, but more recently, Professor Zdeněk Gertz, who is a student of Krzysztof Tarkowski, has established that this actually happens in mammals as well. So it's one of the fundamental principles of development. Once that has happened, the blue cells send a signal to the yellow cells and create another kind of cell type, in this case the green cells. And in this way, the embryo is built up gradually into a whole range of different cell types. So, having said that, I need to emphasize that cell differentiation is a very stable process. Indeed, it's a good thing, because we don't have liver cells in our brain or any other kind of cell in our skin. But the process can be reversed by somatic cell nuclear transfer, cell fusion, or indeed transcription factor overexpression. I would like to concentrate on the first of these because it illustrates a number of general principles. So the content of my talk is this. I'll have a certain amount of background, then I'll talk about nuclear transfer to eggs, and the main conclusion from this was that there is a conservation of the genome. I then want to talk about nuclear transfer to what we call oocytes, and the importance of this is to try to discover the mechanisms of reprogramming. And lastly, I'll make a few comments about cell replacement and the prospects for therapy. We need to remember that the original question in this field was to ask whether all cells of the body have the same genes. And when we go back to the 1950s, you have to remember that we did not know that. It might well have been, and indeed was suggested, that as different cells are formed, they might lose the genes which they would no longer require. So skin cells would lose brain genes, for example. So that was the key question which made people want to do nuclear transfer in the first place. And of course it has current interest in the possibility of replacing aged and diseased cells. And indeed, when the first experiments were done, one has to admit that one had no idea that this current interest in the field would develop that way. So the idea that you should only do research which has an obvious applied medical objective certainly did not apply in this case. Now this goes back a long while, and it shows the story of Hesiod, in Hesiod, where Prometheus discovered the principle, the secret of fire, and Zeus was displeased with that, so he arranged for an eagle, which you can just about make out there, it's the eagle's head, to eat Prometheus's liver every day. And the story goes, and I'll show the original from University. The original says, actually, that the long-lived bird, the eagle, the liver became immortal, and it grew every night by the amount that the eagle had eaten the day before. So it's rather remarkable that that fundamental principle seemed to have been understood 750 years BC. How they discovered it, no one knows, but it's an amusing thought. So the principle of nuclear transfer to eggs is to take the cells from, say, skin, separate the cells, suck a cell into a microneedle such that the cell wall is broken but the nucleus is not, and this can in some cases form a frog, or in more recent work, of course, it works in mammals and can form the adult mouse or other animal. This was the first of such frogs, first cloned animal, going back, you see, to 1958. Now, this was not a trivial problem, because the particular eggs that we were using were those of the South African frog, and it turned out that when you try to inject a needle into this egg, it's covered with an extremely elastic, sticky jelly, such that the needle would go into the egg and it would even come out the other side, still covered by jelly. So it never went into the egg at all. And it was necessary to discover some means by which you could actually inject anything into this egg. That was discovered partly by luck, but it did turn out that you can irradiate the surface in such a way that this problem is cured. Another problem was to have a genetic marker. There was no such thing as a genetic marker at that time. And my boss, Michael Fishburne, as Christoph has mentioned, discovered a mutation called a 1-nucleolus mutation. All these cells have 1 nucleolus, which is different from the 2 nucleoli that other cells have. It provided a genetic marker which was immensely valuable. And the story of that is quite interesting. because my supervisor had a student who was working on a subject like this, and the student came to my supervisor and said, well, you've given me wrong information, because you told me that when a cell has only one nucleus like that, it will always die, because it was haploid. And she said, these cells are not dying. So if I had been my supervisor, like been in that position, I would probably have said what most of you would: go back to the whole experiment again, make new solutions, check everything, and do it all again. And had she done that, she would never have repeated the result. But he was wiser than that, so he said, you can do it again, but make sure that you use exactly the same frog that you used before. Not a new one, the same one. When that was done, the same result was obtained. And that was the history of how it turned out. There was a particular mutation in that particular frog, which happened to give a single nucleus, not two as usual. And it was immensely useful for the whole of the rest of this field. So it was an interesting anecdote regarding the history of how this came about. The next question, once the technique works at all, is whether you can take a specialized cell, like muscle, and successfully transplant a nucleus from a specialized cell. That was really getting to the main question. And it is possible to separate the cells of the muscle. Here is a single muscle cell, and you may see a muscle fiber going right the way through this cell, because that nucleus has encoded or produced special muscle gene expression. When that nucleus is transplanted, you nearly always get embryos like this. Now, this is an embryo which will die because it has cells there, but the rest of the embryo is abnormal because the transplanted nucleus did not manage to divide sufficiently well to populate the whole of the egg with daughter cells. So, is that? Sorry, whether it's further away or nearer. It's more like this. Yes, I'm struggling with this a bit, but I'll let me try it like this. Is that about a bit better? Like, let's hope so. I can't tell whether I'm being heard or not. It turned out that when you take one of these cells and retransplant the nucleus again, you can then get perfectly normal development. There is an explanation for that, but that was how you had to make the experiments work. And eventually one got essentially normal larvae. These are tadpoles. This one from a fertilized egg, that one from a nuclear transplant, in this case from a muscle nucleus. And when you look at the eye of the nuclear transplant from the muscle cell, it's a perfectly normal eye with lens, layers of retina, pigmented epithelium, and so on. But we must remember that each of these cells, several thousand of them, contained a nucleus which had previously been part of a muscle cell. And that therefore showed that there had been a reprogramming or transformation of the muscle cell nucleus from being part of a muscle cell into, in this case, components of the eye. So the general principle of reprogramming was established that way.